this one lah. So, is everyone here? Rachel, 36, 40, right?
For students, it's 39, sir. 39. There should be 39 students.
Yeah. Sama je. I start at 2 o'clock, at 2:30. Sama je. Still short. Still like fashionably late for you. Alright, luckily everyone's online. So hopefully online ni adalah alasan sikit. Oh, connection's not stable and all that. Kalau in physical class, oh sorry, lambat, tertinggal bus. I couldn't remember which bus is it to come to the faculty. Excuses kan. It's normal. Alright, can we, for Muslim, can we start our class with Al-Fatihah? Thank you. Okay, so this week we're going to learn about the protein structures. So we learn about the structure of DNA. We learn about how DNA, the coding of DNA is transcribed and translated into protein. So now we're going to learn about how the arrangement of proteins, the interactions, whatever that is involved within each of amino acids, give rise to a certain shape of folding. That shape of folding, which in turn give the functionality to its protein, either be it enzyme or many other things. Okay, so we are going to look at the function of protein as well. All right. So at the end of the topic, you all should be able to understand and describe the basic structure of protein. Okay. Explain the hierarchy level of protein level. Relate the importance of protein folding to characteristic of foods. So we're going to discuss about that if we have the time, hopefully. So protein, it makes up for about 15% of the cell. Is it a lot? I don't know. You tell me. Is it a lot?
No. No.
How do you know? It's not a lot. Because it's 15. Yeah, it's not as much compared to your carbohydrates and your fats. Okay, majority of our cells, okay, majority of them is made of by fats. The one conquering, because fats involved in your making of your cell membrane, same as your carbohydrates. Okay, so what's the function in the cell for protein?
It provides energy.
Okay, it provides energy. Okay, what else?
Active transport.
For transportation. Okay, yeah.
Repair the body tissue.
Repair. Okay, recovery and repair. Okay, what else?
It acts as an enzyme to increase the rate of chemical reactions.
Exactly. One of it is function in biochemistry process, act as enzyme. Lagi?
Maybe like signal or markers for like cellular identification.
Correct. Because they cell to cell, they cannot say hi to each other, right? The only way they can communicate is through proteins, send signals and sensory. Good. What else? Structural support. Yep, betul. Thanks, Mufliha. Structural. Might rosak. Yeah, structural support. Do you know where it has a structural support? Kat mana? Where is
Our
it?
hair, sir. Keratin.
Yeah, keratin. But in terms of cells,
Cell wall.
hold on. Okay. In terms of cells, it regard to scaffolding within the cell membrane. If you notice that, if your cell membrane too, some of it is embedded of your protein structures. Some of the protein structures is like I think seperti Ilman maybe said, is for membrane pores and membrane channels. Okay, membrane protein pores is macam gate lah, macam UPM certain gates, and the rest membrane is just
Doctor, you're lagging.
Wall. Okay.
Doctor, you're lagging.
So these are lagging. Am I lagging now?
Yeah, still lagging. Your video is not working, so you're like stuck at one motion.
So actually, this
Afia, are you recording?
Hi,
Hi, Doktor.
Doctor.
you all hear me?
Hi,
Yeah.
doctor.
Yes, Doctor.
Okay. Sorry about that. I think my internet is giving me some sort of revelation, kind of rebel today. They don't want me to teach. It's a Mac. Okay, hold on. PowerPoint. Okay. So am I still lagging now, or am I smooth as ever?
Much better,
Smooth.
doctor.
Much better.
Okay, cool. Right. So here are the some of the functions of enzyme of protein in the cells. Okay. So like I said, majority of the enzyme protein are being used as an enzyme. So most of your chemical reactions, or I'm not sorry, most all of your chemical reactions are being controlled by enzymes. Okay. Structural. Okay, these are the ones that hold the integrity of our membrane, cell membrane. Okay, like I said, they act as scaffolding. Scaffolding ni kalau tengok orang buat rumah, it's like tiang rumah or macam cerucuk. They call Malay called cerucuk, right? These are the scaffolding that hold the structure of the house together. Okay, our cell membrane has that too. Okay, without the scaffolding of the protein membrane, your cell wall is like macam amoeba, like gooey, like it can shape like kind of like that. It changes shape whole time. It doesn't have permanent shaping, right? Transport, of course, it carries nutrient, it brings nutrient, it um, eliminates nutrient. Motors for what? For movement. Okay, this is not only related to human cells, any kind of cells from living microorganisms. Storage, it can store nutrients or other signaling can produce. Signaling and receptors is same category. They are for communicating. Gene regulations to control transcription of your genes. Okay, whether to turn on or turn off. All right. Defense, yeah, of course. Any immune system is being created by, is being controlled, is by your protein, and other special functions. There's a lot of other special functions for protein in terms of controlling the growth, right? Replicating and everything, all that. Okay, so protein structure is important as the shape of the protein allows it to perform particular role or functions. How this thing, how when I put these two images together, how this is related? Teka-teki, ding, 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 ding.
Splicing.
What? Splicing? Kind of. Okay, what's the function of splicing? Boleh lah.
Rearrange some codes of gene
Rearrange some code for what particular things?
to create multiple different
Okay.
kind of proteins.
That is during the transcription, right? What about, you imagine the string itself, right? The benang ni, the string itself is a chain of your polypeptide. So what's the function of scissors?
Doctor, is it related to the Cas9 protein thing?
Is it related to what, Ilman?
Cas9 protein, the one in CRISPR, is it related to that?
That one is a bit advanced. I'm just asking a bit simpler in terms of your protein structures. Okay, like I say, if you look at the description that I just read to you, okay, protein structure is important as the shape of protein allows it to perform particular role or function, meaning that if satu strings of protein like this, right, satu benang saja, do you think it's functional? Like only a string, satu benang saja of polypeptide, do you think it's functional? No? Okay, why no, Sharifah?
Maybe sebab kalau satu string tu macam dia tak cukup coding.
Dia tak cukup protein?
Coding.
Tak cukup coding? Not really. All the coding is already in one strings. So if you know the shape of protein or enzyme, how does it look like? Is it like one string? Or is it like apa dia? What does it mean by this?
Bergumpal.
Gumpal, folding. So your protein is actually, when you saw the pictures, your protein is actually originally is that one string of multiple amino acid that being arranged together based on the coding of RNA, correct? One string, right? After that, what happened? It needs to be functionalized, isn't it? It needs to be folded so that it become functional. Pernah ke jumpa protein yang satu baris je? Tak pernah kan? So it needs to be folded in a certain way, macam origami, right? Like a piece of paper is your polypeptide. You need to fold it in a certain way so that it become a boat, it become an aeroplane, it become a duck, or become flowers, anything that you wanted it to be. Okay, so the arrangement of proteins, the arrangement of your amino acids, when I say scissors here, so this has to be related to how you cut and then how you modify the strings of polypeptide, making it to a certain shape so that the shape will be a functional type of protein. Faham ke? Okay, once we move on, I think you understand much more better. All right, so I think you heard this and you saw this so many times. Protein has 4 different levels of conformation. Each of these serve as different function. Okay, it's the first one. The first one after it really finished the punya translation, is just an erase, straight erase one string of macam benang tu, one string sequence of your amino acids. It's a polypeptide. Okay, and then you have secondary structure where you have minor folding. You have tertiary structure where you have secondary folding, which folding further than that, and then you have quaternary structure, whereby the combination of multiple type of polypeptide. For 1, 2, and 3, this 3 is only regards to single polypeptide, satu chain, satu arrangement of one whole chain of polypeptide. So when it comes to quaternary, there's a possibility whereby combination of different polypeptide that comes together in addition with other types of compounds. Okay, so let's look at and move on. Okay, so there are 20 different amino acids, but they can be joined together in many different combinations to form diverse range of protein. So basically, the one that separates them is just R group. The rest semua dia punya base semua sama saja. Okay, sama kalau macam you buat any kind of houses, kalau buat rumah, the base of rumah semua sama je, but the roof, the door, the windows, everything is different. The differences from your R groups. Okay, let's look at these. Okay, so these are the base of your amino acids. Okay, this one is the middle carbon. Okay, the central carbon, and you have on your right hand side is your carboxyl group, which is acidic. Okay, which the loss of H will make this negatively charged. And then you have amide group on your left hand side, which is alkali charge, which the loss of this H, if in aqueous form, it can become plus group. So your R chain is different here. Ini saja yang berbeza. Okay, for the first one, if I'm mistaken, this one is alanine. Okay, amino group, acid group. So this this is alanine. Okay, but you have a side chain of another carbon, alanine. Okay, and you have valine, where you have addition of other 3 carbon on your R group. and you have cysteine. You have sulfide groups. Okay, here, and then you have phenylalanine, where you have aromatic group. So the difference of all these is just we call it R group. R group, the ones is varies lah. So these are the one that determine the differences between 20 amino acids. The rest sama saja. The base semua sama saja. Okay. So this is why you call aliphatic or bases if your amino acid, once your amino acids in aqueous form, because the aqueous form, this will be released, right? Become plus amide group, the amino group become plus, this acid group become minus. Okay, clear? All right. So this is a group of amino acids. Yeah. So amino acids, you have two different types, two different categories: non-polar or polar amino acid. I'm sure you know the difference between non-polar and polar. Okay. You know what's the difference between polar and non-polar? Tahu kan? Still remember? Polar and non-polar. So polar is non-polar is the punya electronegative charge is equal. Okay, between the two sides, between all of these compounds or atoms within your compounds. For non-polar means that the polarity of one side of the group is more polar or more charged as compared to the other. So for polar amino acids, okay, for non-polar, I think ada 1, 2, 3, 4, 5, 6, 7, 10, 10 kan, okay, 10. For polar acid, is divided into 3 parts, okay, whereby the first part is uncharged polar. Uncharged polar ni nanti we tengok the differences, and then we have positive charge, negative charge. Okay, for the positive charge, for the positive charge, this positive, sorry, for the positive charge and negative charge, these are all contributed by the R groups. Okay, so let's look at it. Okay, this is called non-polar. All right, so this is the side group is called non-polar. Everything is just balanced out. So supposedly we have, okay, they have 9, and they have polar non-charge. Okay, if you look here, the minus and the negative, okay, this one checks out, balance each others. So this is uncharged, right? The R group is uncharged. Okay, and then we have negatively charged amino acid, whereby the R group is negative charged. These are all conditions whereby in an aqueous form, yeah. Okay, and then finally we have positively charged. Okay, so basically, what I want to highlight here is not, I don't expect if you can hafal, that's very good lah. You have a very good photographic memory, but I really don't expect you to hafal and then to know each of this compound structure. No need. All right, we're not. There's no point for you. I asked you to hafal because we're not testing about your memorizations. Maybe some of you are very good, some of you are not. We are testing a bit based on your understanding and your critical thinking. So no need to hafal, but just understand why is it positively charged, why is it polar, and why it's non-polar. Okay, what contributes towards that. Okay, so the reason why I explain all this, meaning to say that each of the amino acids has different kind of interaction, have different kind of charge, right? Kiranya dia punya interactions and dia punya repulsions, attraction and repulsion, is different one from one to each other's. So meaning to say, same as humans, if I were to have 20 people or 20 students of you, 20 people of 20 students of you guys, I don't really expect one person to the other are similar to each other's. Very unlikely. Surely most of the time, one person to the other of all the 20 people, surely they have different attitudes, they have different mindsets, they have different characters. Male and female, doesn't really matter. So all of you guys are different. So based, like I said, when doing the first class of DNA structures, same as DNA, the interaction between DNA, the interactions of amino acids is going to be different. For DNA, you bayangkan, for DNA, the shape as it is, it hardly have 4 different variations. A, C, T, G. That's it. So the structure different, the structure punya variation is not that big. Okay, the interaction is not that big because only have 4 type of people. However, when it comes to amino acid, you have 20. So bayangkan ada ragam 20 orang, how they are attract to each other that can relate to their punya structures later on. Okay, macam cakap dulu lah, if I were to put you guys 20 amino acids in a class together, I'm sure some of you guys will stay close to each other and some of you guys will stay away from each other due to your interactions of repulsion and attraction. Make sense? That's what I want to highlight in this structural difference of your amino acids that later on will give the interactions. Okay, so I'm sure you look into this. After you understand about this, this is assembly of protein whereby each of amino acid, this happened during your translation, yeah. Okay, with the form of energy, ATP energy that transfer from one amino acid to the other. At the same time, your peptide bond is bonded through the release of hydrogen, to the release of water. Okay, so it grows based on your addition of amino acids. This is what I call condensation reaction. Condensation because they release water. Okay, this is very simple lah, straightforward. Sama juga ini. Okay, so this is called peptide bonds. Okay, your alpha carboxyl group of one amino acid are joined to alpha amino or the second amino acid. They release water. Okay, sama juga, you nak break the peptide bond, you insert water. You want to combine the, you want to make the peptide bond, you release water, right? Hydrolysis. So only alpha carboxyl group and alpha amino group are used, not the R group. So in any words, at any time, your R group ni memang takkan involved in your making of peptide bond at all. Only your amino group and carboxyl group saja involved. Your R group will not be joining the interactions. They're just freely on the side chain. That's it. Okay. But the ones that usually have this kind of interaction is your protein folding is your R group. Okay. So these are called local folding of fluid into regular patterns, which is your secondary structures. Okay. Regular pattern is pattern yang very basics lah. Okay. So the first one we have this forget about this. You don't have to focus about this. It's very complex of Ramachandran plot. How this Ramachandran plot give you in terms of alpha left hand alpha helix, right hand alpha helix, beta sheet. This is all regarded of the conformation of your amino acids. Just forget about this. Okay. All right. This I want to focus on. So right-handed helix, yeah, the carbonyl oxygen of residue I, okay, meaning that one of your oxygens based on the carbonyl group or carboxyl groups form hydrogen bond with the amide residues, okay, in amino acid number 4. Kiranya 1, 2, 3, 4. The carboxyl group will combine with number 4. Let me quickly draw to you. Yep. Okay, like this. Okay. So when I say right hand helix, meaning that these are clockwise turn, right? So if you want to imagine how your helix looks like, it's just like your thumb, and you twist it like this ke dalam, so it goes like that, Yep, macam ni. So let me quickly draw. Yes, what's my drawing? Hold on. Okay. Sorry, my crap drawing. Okay, kind of like that. Hold on, yeah. I think I lost. Sorry, my computer be hanging. Stop showing. Okay, let me do it again. Sorry. Let's say it's draw. Okay. All right, kind of like this, yeah. It's clockwise. Okay. So you have your carboxyl group of oxygen. So there's a number 1, and then you have number 2 of your amino acids, number 3, and then you have here number 4. Your amide group. So these are the one that joins together. Yeah, that makes that whole and then creates that spherical shape. Yeah, so it's very simple. So interaction is always your carbonyl oxygens with your hydrogen bond for your amide residues, your amide, your amino group lah. Okay, so the propensity of a peptide, hold on, keep hanging, undo, stop, okay. All right, sorry. So the propensity of a peptide for forming an alpha helix depends on its sequence. Okay, nanti I'll explain to you. Okay, proline does not fit into helix. Okay, this is called proline. What the amino acid is proline. If proline tu ada dalam you punya amino acid sequence, right? Katakanlah dalam tu ada glycine, alanine, cysteine, what else? Asparagine, aspartate, and everything. Suddenly within that ada proline. Amino acid called proline. Ada proline je, they can break the whole system. It won't become continuous helical. Maybe if I'm mistaken, proline ni is they call that helix breaker, because proline ni can make your, instead your helical go like this, dia kan ada abrupt turning, macam dia terkepak. Nanti we'll look into this, yeah, the structures. Just keep into your mind first. All right, this is beta sheet. It's called sheet because it's literally macam a sheet of papers. Okay, for your amino acids, it can turn and then it can become side by side. Jadi a piece of paper macam ni kan. It's all like one single. Here is like one single of your amino acid, one length. It can be folded like that, macam ni. So these two, which is actually is a single polypeptide, can be arranged like this or bonded side by side. For helical, they're bonded by up and down. For beta sheets, it is side by side. Okay. So this second structure can be either anti-parallel like this, okay, or parallel. Okay. When it comes to anti-parallel, if you look into the R group, yeah, the R group is one of it is upside, one is the other side. Okay. So it's not, sorry. Okay, betul. Right. So basically, one group of R group is at the bottom, this one at the top. Okay. So the bonding of hydrogen bond between carbonyl oxygen and amide groups is also hydrogen bond is equally parallel. Okay. These are called anti-parallel because one face up, one face down. Okay. So sama juga the propensity, the propensity the what called propensity tu macam, how to say, not the adherence, propensity. Okay, propensity tu macam, if you don't understand, propensity tu macam what are the words? Can someone help me? Propensity.
Tendency.
It's like, how to say in Malay? Dia punya,
Menaikkan.
Sorry, apa dia?
Menaikkan. Macam nak?
Menaikkan. Bukan, dia macam dia ke
Setiang
arah
tu.
situ. Bukan setiang, bukan. Propensity ni macam dia macam kegalakan.
So is it the inclination?
Cenderung ya, Doktor?
Inclination?
Yeah, inclination.
Cenderung.
Thank you for the inclinations. So the inclinations either toward the beta sheets or toward the alpha helical. So like I said, the arrangement of your protein, the arrangement of your amino acids is inclined towards what? So all that is depending on what type of protein, what type of amino acid you have within your sequence. Nanti we look into there's one table I'll show you. You will understand much better after this. Okay. So this is from the side view. Okay. If you look at the side view, the arrangement of R groups is kind of like that. One at the bottom here and one at the top. Let's look into the parallel. Okay. Macam parallel ni, if you look the R groups ni, so this is, okay, this is anti-parallel, this is parallel. Okay, this one, okay, betul. This is anti-parallel sama now, and you have entire parallel. So they have, sorry, this is entire parallel, this is parallel. Right, sorry about that. So if you look at the parallel, the arrangement of R groups sama saja. Yeah. There's no crossover between. There's no up and down. Everything is facing down or everything is just facing up. All right. So the hydrogen bond is slightly crossed, not directly parallel. Okay. Regardless, all this has, if I'm not mistaken, all of these has between parallel and anti-parallel, the strength of bondings is almost equal because the one that holding it too, it's almost similar. So if you look here, this are ini macam top view. If you tengok dari atas, the sub view is like this. So you punya, like I said, if you remember, you punya polypeptide tu is like one string of your DNA. They can be folded, U-turn like that, okay, and they can become a form a sheet. Yeah. Okay. These are the examples of anti-parallel and parallel. Okay. So anti-parallel, the arrangement, the bonding of hydrogen bond, your O with amide group H, which is side by side. And then you have a parallel, which is crossed like this. But the R group, if you notice the R groups, almost this is R group, yeah? Here, same. For this one, that's upside, one down, one facing down, one facing up. Regardless, more or less it's the same, but the arrangement is a bit different. So if you look into this, when I say the arrangement is different, because if you look here, these R group are facing each other. So they have different kind of interaction and bonding. As for this one, this one more or less are away from each other. So all this has, when it comes to higher level of hierarchy structures, it's gonna have different foldings. Okay. If you look into your structure of protein, it's like domino effect. Small changes would affect the biggest one. Okay. So even one single amino acid can affect the highest level of protein folding. Nanti we'll see how is it related, how is it affected. Okay. So this one, beta turns in the beta sheet allow the protein backbone to make abrupt turns. Okay. That's why they can be side by side. Okay. So it can have a turn. So all these turning beta sheet and alpha helical shape, again, the propensity of a peptide forming beta turn depend on its structures, arrangements of sequence. Hold on, yeah. Okay. Okay, so these are, if you look into the 20 types of amino acids, alpha helix and beta sheet and beta turn element distribution among amino acids. Hold on, guys. Sorry, my kids driving me crazy. Sekejap, ya. Okay, sorry, iklan. My kids are driving me crazy. Sorry about that. Okay. All right. Okay, these are very important. Okay. So if you look at this, we have like 20 different types of amino acids. So if you look dia punya strength ni, okay, dia punya level of strength ni, okay. If high, kalau you nampak more merah, that means it's stronger lah. If so, you have glutamine, you have in your sequence, yeah. If, for example, you have glutamine, methionine, you have a lot of glutamine, methionine, alanine, what else? Leucine. Your shape is tend to be more helix. Yeah. However, imagine if you have glutamine, methionine, alanine, but like 10 to 15 amino acid sequence, and then suddenly you have proline, you have here, look here, proline, glycine, asparagine. What do you think happened? It become like this. Contoh let's see. Okay. Contoh. So the first part you have methionine, a lot of glutamine, methionine, alanine is become right-handed like this, yeah. Slightly within that sequence, ada proline. Proline is known to be a breaker of alpha helix, or they can make B turn. So it becomes like this. Faham tak? Okay. And then suddenly, alamak. Okay, hold on. Okay, sorry about that. Let me try again. Okay. Awal dia macam helix, ya. And then you have proline, they make abrupt turns, and then after that you're gonna have leucine, valine. Okay, these are the ones. Oh my goodness. Oh, sorry, my computer is driving me crazy. Stop share. Share again. Okay. All right. Again, I try it again lah. Hopefully you understand this time. Here, okay, you have mula dari alpha helical. Okay, and then you have proline abrupt turn. Okay, and then you have beta conformation means that these are the one that makes the beta sheets. And then you have proline again, they make abrupt turn. You have the glutamine again, they become alpha helix. Faham tak how the arrangement works? Is it clear to you guys? So basically, all of these, right? If you see the strength, okay, glutamine and all these, they are most likely will fold it into alpha helix. Okay. For here, methionine, leucine, valine, if they were to arrange closely to each other, most likely they will form beta conformation, either beta parallel or anti-parallel. If within your amino acid sequence, you have asparagine, you have threonine, you have aspartate, asparagine, proline, and glycine, it suddenly make a different turning. Okay, all these will relate to how your structure works. Okay, nanti we'll look into your tertiary structures. Okay, move right. So these are called beyond secondary structures. Okay, this secondary structure can either be anti-parallel as shown, or parallel and need not to be planar. Flat lah. Kalau planar is just flat like this. It's not flat. It's like a curve of up and down. Okay, as shown, but it can be twisted. Okay, the propensity of a peptide forming a beta sheet depends on its sequence. Sama je. Basically, you really have to understand this, right? All of the structures, folding of structures, all depend on what you have within your polypeptide sequence. Okay, so let's look into your tertiary. Okay, tertiary sama juga. Tertiary is just another interaction within your secondary structures. Okay, so this is called global folding or protein chain, final 3D structures of your polypeptide. Okay, this is folded into native functional form. Tertiary structure can stand on its own, or they can go into quaternary combined with other subunits. Okay, tertiary structure ni pun dia boleh work as a functional protein, or it can be part of bigger protein. Okay, so these amino acids far apart in primary structures may be close in tertiary structure. It's like this. Okay, your secondary structures is like your primary structure is like a single individual of you guys. Secondary structure is like you make into each groups. Okay, you are the group A, group B, group C, group D. And when it comes to tertiary structures, is how each groups interact with each others. Okay, maybe group A dengan B suka each other, or because diorang sama hostel, sama dorm, or maybe A dengan C tak suka, or dia ni musuh, rival dengan this guy, this fella rival, ataupun this guy pernah break up with this girl. So the group is like it's not like not mixed up with each others. So it's like how each of these group interact with each other in a bigger scale lah. Right? So like I said, each amino sequence give rise to your secondary structures. Your secondary structures will give rise to tertiary structures. That's why I say it's domino effects. Yeah. Okay. So for tertiary structures, okay, if you learn before this secondary, only hydrogen bond that is being involved. For tertiary structures, more than that is involved. For example, you have ionic bond, okay, which is strong bonds compared to hydrogen bonds. Okay, the interaction of different electronegativity, plus and minus. You have your disulfide bond. Okay, the bridging between sulfide and sulfide, which is a very, very strong bond. You have hydrophobic bonds, whereby these are the groupings of side chains of your hydrophobics. And you have hydrogen bonds or dipole bonds, and you have ion dipole. Okay, ion and ions. So all of these foldings, okay, is interactions and repulsion from each others. Okay, for example, katakanlah macam hydrophobic. Yeah, what is hydrophobic?
Takut air.
Okay, good. Takut air. Afraid of water. So basically, if you were to have your protein ni, right, you campak dalam air. So suddenly, this group of hydrophobic will tend to stick to each other and stick to the middle or core parts of your protein. Protected lah, macam your micelle. You still remember your micelles? M-I-C-E-L-L-E. Micelles. Okay. Look at your chats. Micelle. Yep. So micelle ni, outside part is non-hydrophobic, and inside part is hydrophobic. So hydrophilic. Okay, hydrophilic and hydrophobic. Yeah.
Is it like the phospholipid bilayer thing?
Kinda. Yes, correct. Kinda. Same. So if interactions between each other, so regardless if it's with water or anything, all these are folded the protein in a such unique way. Okay, like I said, and I've been stressing so many times, the propensity of protein structure or your tertiary structure or secondary structure depending on amino acid sequence. What sort of amino acid sequence within that? Okay. For example, katakanlah, okay, the easiest way I could explain it and put into words, whereby I have 20 people of you, 20 different kind of people, and I arrange them between, I give them numbers, right? 1 to 20. Okay, 1 to 20, they form like macam, katakanlah macam zigzag shape, yeah. So the moment I say, okay, go randomly. Now we have random number, like the arrangement is like 1, 3, 5, 6, 10, 11, 12, 13. Now the arrangement is different. Do you think it's going to be a zigzag?
No,
Why it's not going to be zigzag?
because of the bonds between
The bonds
the
or the interaction
molecules.
between each others. Like I said, characters of humans sama juga. Protein ni sama juga characters of humans. Some of you guys might not like each other. You may become zigzag, some maybe zigzag, zigzag, zigzag. Suddenly, ada buat macam U-turn and then become straight line. Okay, that U-turn maybe ada differences between between some of you guys, ada macam fight with each other or you repulse each other. You tak suka each other. Oh, tak suka Mamat ni. So you tend to stay away from each other. So the arrangements, although you have 20 same people, but once I move you all randomly, then you punya shape's going to be different. Does it make sense to you guys?
Yes.
So it's all about arrangements. Regardless if your sequence tu sama je semua, 20 tu sama je orang, tapi how I order you guys or how I arrange you guys, they're all going to give to a certain unique shape randomly. Yeah. This one bukan paksa-paksa kita, okay, kita nak attach dengan ni, dengan ni, dengan ni. No. It's just how they comes together based on their interactions. So I can't I can't stress this thing heavily enough how important it is to have to protein structure. It is depending on amino acid sequence or order. Okay. All right. This is tertiary structures. They call it super secondary structures or motifs. All right. Beyond secondary structures, small, discrete, commonly observed aggregates of secondary structures, repeating patterns of secondary structures. All right. So you have beta sheets, all the way beta sheets, or you have helix, and then you have turn looping, and then you have helix, or the combination of beta, alpha, beta. These are called much simpler patterns or repeating patterns. When you have domain or foldings, okay, domains or folding is independent units of structures. meaning the combination of these secondary units folds into different kinds of shapes. It's not repeating. Okay. For example, you have beta barrel or 4 helix bundle. Macam ni ya. Common motifs. Motif ni simple repeating. For example, helix loop helix. You have helical here and then you have turn and then you have helix. Okay. Coil meaning the combinations of your helix comes together. You have helix bundle, whereby you have the combination of helix all together, whether right hand helix or left hand helix. Right? Okay. Another example is beta alpha beta. Okay, you have sheets here, looping, helical, sheets. You have hairpin, whereby you have sheets and sheets, and we have beta meander. Okay, you have sheets all the way. You have Greek key, beta sandwich. Okay, this all just simple repetitive of your secondary structures. When you come to folds, yes, you have alpha helical, but they are in more complex way. They are folded in a certain way based on interactions, complex interactions, right, of tertiary, whereby you have not just simple shape like this, but it tend to be some sort of into a certain shape. For example, twisted sheet, like this one. This is beta barrel, macam like a barrel lah. This one's alpha beta barrel, combination of alpha helix and beta. That makes some sort of like a barrel, kind of like a baldi. And then you have beta helix of conformation like this. Okay. So in terms of differences between these two, your folding are much more complex. Yes, its own structures. Okay. As compared to your motif. Motif is just repeating of your secondary structures. All right. Quaternary. Let's take a break. We take a break for about 5 minutes, and then 5 to 10 minutes, then we come back. Let's say we come back at 3:30. Is it okay?
Okay, doctor.
Okay, Doctor.
All right. Do you have any questions before we take a break, or you want to ask afterwards? Okay. So we take a break first. If you have any question, just type in the chats. We can discuss it later. All right. I'll see you in about 6 minutes. Yeah. See you guys. All right. So you have any questions? Ada soalan tak?
Doctor, I have a question about the structures. Like you said, the beta helix and there's the sheet one, right? The beta sheet shapes, right? Would it like affect other proteins like to become their shape?
Sorry, what was your question? The beta helix and alpha helix, will it affect the shapes? Is that what your
Or like,
question?
just now you said like ada shape yang helix, ada shape yang macam paper tu kan?
Mm-hmm.
Would like the paper punya shape tu,
Yeah.
kalau dia interact dengan protein yang helix, so dia akan change the shape tak?
Of course, I'm not saying they change, because you're not, change means you are adding something or you changing something. That means it changed lah. If you say it's a native form whereby you have helical and then you have sheets side by side with each other, that will affect how its arrangements. Okay, like I said, there are lots, and the one example that I showed to you is just some example that exists within the tertiary structures. But that doesn't mean if they have helical here, one helical here, and one sheet here, doesn't mean it could be side by side. It could be further part away or much closer or interact with each other like that. That's all depending on what sort of amino sequence within that. So if you remember, you punya tertiary structures here. Okay, this one. Okay, so they call another folding of another interactions apart from your secondary. Secondary is just hydrogen bond, right? It's within the polypeptides, among the
Yes.
polypeptides.
Yeah.
These are the R groups. If you look at secondary, only your carboxyl group and amide group. So when it comes to tertiary structures, this involve you punya R group. How R group relate to each other. So does not mean if helical here and helical here, it could be only like this or only like that atau straight macam ni. It could be like macam, it could be like cross like this, it could be further apart. Macam ni kan? Here you go. Okay, like alpha helix pun that does not mean necessarily it can be side by side. It could be looping away from each other. It could be coiled within each other, or you have helix bundle. They're all depending how the interactions within that. More complex folding, other than you punya motifs, okay, it could fold in another kind of shapes. That's all, like I said, that's all depending on the interaction between the R groups. Clear, man?
So like if ada different kind of macam interaction or something like something full or cross macam tu, it would give out different functions lah kan?
Yeah, for sure, for sure. Like I said, I can't stress in terms of this particular sentences or words. Here you go. The propensity of a peptide forming beta sheet, alpha helical, or any kind of structures, depending on its sequence. Okay, this one, this one. This I don't expect you to hafal. Really, I don't. Okay, but you have to understand how arrangement of all this. For example, if I were to give you a certain, okay, glutamine, alanine, you have this kind of table. What do you think the propensity or the inclinations it will perform towards two? Is it alpha helical? Is it beta conformation? Or is it beta turn? So you look like kat sini. Mana dia macam it headed towards that formation. Okay. After dah finish secondary, then baru ada interaction between the R group. Faham ya? Anyone else have their questions?
Doctor,
Yep.
what if the protein is misfolded? What the shape?
What is the what? Sorry?
Apa? Misfolding protein.
Misfolding. Okay. Protein somehow protein won't misfold. Okay. We only say that it's misfold because due to a certain sequence that supposedly is not there. Supposedly it, okay, katakanlah original or native form, original dia akan fold like that. But due to a certain amino acid sequence, one or two, either if it's only one point, suddenly if supposedly it's shaped like that, it's shaped like this ataupun shaped like this. Bayangkanlah je, supposedly it's globular like that macam ni. Now ada one or two sequence or even one sequence, it's going to shape like that, it's going to shape like that. Because it's like katakanlah you ada 20, okay, you ada 20 people. Okay. One of them is me. Okay. I'm the one who lectured to you guys. I'm the one to give lecture to you guys. Suddenly, instead of me give lecture, ada orang bagi lecture. He or she may be better than me or maybe less better than I am, depending. If it's better, maybe you all macam, oh, lagi happy, lagi get together, lagi fun, korang pergi jalan-jalan sama, like, for example, you are more closely related to each other. What if orang yang datang tu, dia ni suka marah-marah, like, herdik-herdik, maki-maki korang, they break up the whole group, right? You tend to, malah dalam kelas ni, ada seorang maybe like, instead of 19 people come, only 10 of you comes, ataupun instead of like interaction, you rapat dengan this lecture, you tak rapat in this, you just hated this guy, right? So interaction between these people are different. So the changes of one amino acid sequence will affect the whole structure. Faham tak? So they call misfolding. Misfolding is due to supposedly orang ni yang masuk dalam that group, tapi bukan orang ni masuk, orang lain yang masuk. That will cause misfolding. Misfolding ni supposedly dia lari from its original or native form. Faham tak?
Okay, sir.
Okay. Clear? Anything else?
So doktor, yang doktor
Ya.
kata misfolding tu, kiranya dia akan ubah structure lain lah macam tu kan doktor? Dia akan jadi berbeza
Different
lah.
shape. Different shape. Nanti I give you example. Just keep in mind, simplest example will be anemia sickle cell. I think I explained it to you last week. Whereby you punya hemoglobin is like biconcave shape, like dua piring attached together, macam ni, right? Now it's become, instead of biconcave shape, it become flat sickle. Macam ni. Ya. Nanti tengok. Okay. Alright, let's move on. Nanti nanti kita have another chat of this session. Okay. So common examples. Alright, so you have tetramer. These are the bindings when it comes to quaternary, okay, this is the highest order of assembly of protein, the highest order, whereby you have combination of different subunits. One subunit is from tertiary structures. Okay. Subunit are held together by weak interactions. Okay, between all this is maybe hydrogen bond or hydrophobic bonds. Okay, it's not as strong as your tertiaries, which is with each others. Okay, this sometimes give the quaternary structure some sort of flexibility. Okay, maybe it can change shape. For example, macam contoh I would like to give example of your red blood cells, whereby the moment it carries oxygens, it will change some sort of shape. Okay, to tell that because it have limitation for your hemoglobin, it has limitation to carry amount of oxygens. Once it's empty, once it's full carrying oxygen, it has changed shape so that no more oxygen can get through. This is due to its weak interaction. It can have a sort of flexibility. Dia punya bentuk berubah sikit lah. Faham ke? Contohnya macam, how to say this? Macam you carry bakul yang penuh dan bakul yang kosong. So bakul yang penuh, it means that you punya bakul tu, or plastic bag lah, say plastic bag, just sumbat all your foods or all your stuff within that. So no more of your barang-barang lain tak boleh masuk dalam plastik tu dah, because it's just full. Your plastic is now from kecil-kecil kerompol, you dah masuk barang, it become big, it change shapes. This is due to weak interaction between your subunits. Okay, that allows it. Faham ya? Okay, these are common examples. Tetramer, but this is all bentuk lah. Tetramer is your single strand binding proteins. Okay, this is during your replications. You have your hexamers with the hexa. Hexa is like 6, right? Okay, 6, 1, 2, 3, 4, 5. Yeah, hexamer. Okay, this is DNA helicase. So DNA helicase tu apa kan? During your replications tu. Okay, during your replications, this helicase is used to unwind your DNA structures, and then you have your filament. Recombinase. Recombinase is to join side by side or adjacent DNA structures. Okay, all that is combination of different subunits of your protein. At the same time, it can be added other compounds, not just proteins. Okay. So this is called hemoglobin or hemoglobin, a molecule to breathe with because it has the ability to carry oxygens. Okay, here are the heme attached to atoms, iron atom, and whereby you have your combination of beta globin and alpha globin. Okay, alpha and beta is just different arrangement of globin. Your globin is your protein lah. Okay, your polypeptides. If you look at here, it noted that sickle cell mutation. This one some beta globin, meaning that one different amino acids in your beta globin will change the whole instructions. It's like I said, domino effect. You change one, it affect habis dia affect semua, kacau semua. Okay, kalau Melayu cakap satu titik nila rosak sebelah. Lebih kurang macam tu lah. Satu change dia is just like it ruin every single thing. So that's the reason when it comes to your DNA structures, when it comes to your transcription, when it comes to translation, folding, everything is just domino effects. It has to be accurate and specific. Okay. So, all right, iron atom. Iron atom are responsible heme and the ones that hold your iron atom. Iron atom ni lah the one that responsible to bind with your oxygens, carry oxygen and carry your carbon dioxide. Okay. Okay, these are the each protein, the sequence of amino acids for your its applicant is for your beta globin of your hemoglobin. Okay, if you look here and LKTDW and everything, it's not, this is not I randomly type and it's not, yeah. If all of these letters represent a certain amino acids, you can Google it. I can't remember, I don't memorize it. But if I'm not mistaken, for a beta globin in your hemoglobins, okay, in your red blood cells, I can't remember which one, but one of these, yeah, the third line, one of these, only one changes of amino acid sequence can lead to sickle cell. One je. Nanti I try to find the one that's different. I can't remember. I think one of these. Yeah. Only one je. Daripada you punya, from your shape of your red blood cells, like a concave biconcave shape like that, right? It becomes flat like this. Okay, so much different. It's only due to one single culprit of amino acids. This is what I call misfolding lah. Okay, one changes that can cause misfolding. Misfolding means that the fold of protein structures is not original or native, or what it's supposed to be. That's called misfolding. Salah lipat. All right. So this is an example of coordinate structures of or non-polar R groups. Okay, your non-polar. So non-polar R groups tend to be buried at the cause of your soluble proteins. For example, macam ni, if you were to fold it, okay, your protein you fold kan, and then when it's in aqueous form, it tends to fold it a certain way whereby your R group or your hydrophobic group tend to stay core or the soluble protein. Macam micelle lah, atau macam apa Ilman cakap tadi? What?
Phospholipid bilayer.
Lipolipid. Okay, yes, that one. Okay, so anything that is hydrophobic, they will tend to stay the blue one, the blue color. They'll tend to stay inside. Okay, and those that are polar or hydrophilic, they tend to stay outside. So basically, this one also, aqueous form and non-aqueous form, that one also some sort of influence, yeah, influence the shape of your protein structures. Okay, the more ada interaction of water je, so the one yang takut air dia akan stay dalam, the one yang suka air dia akan stay kat luar, protected lah. So that also give the certain shape towards your finished product. Okay, sama juga macam your cell membranes, whereby the polar groups, they tend to stay exposed outside this cell membrane. Whatever the inside is this all the groups of your non-polar. So protein, okay, there are two types of, I think from there are two types of protein that is embedded within your cell membrane. One of them is your scaffolding, like I said before, the one that gives structures to your membrane. Yeah. The other one would be your protein channels, whereby the R groups stay outside, the R group of polar will stay outside, creating some sort of pores, and your hydrophobic, right, or your non-polar will stay inside. They are creating a channel so that a certain form, a certain compound, nutrients, or you want to eliminate waste or toxics. Okay, this through all these channels, because you punya cell membrane is pretty tight. It's really like a wall, benteng lah. Okay, like most of your compounds cannot pass through. Okay, so you have to use protein channel like this. Okay. So membrane protein have adapted to hydrophobic environments. Okay, this is due to the certain folding lah. Siapa suka air duduk kat luar, siapa tak suka air duduk dalam aqueous form. Okay, that's it. Done. That's about it. So basically, just recap together. Protein are the key players in our living system. Why? Why they are key players? Although they're only 50%, kenapa they key players?
Sebab function dia, doktor.
Function dia? What function?
Like the enzymes and the structure, all that.
Exactly. So anything that's regard to your biochemical process, biochemistry process within our body, that make your body function, makes your living organism function, your food functions, plants and everything. So basically, they are the one that controlling and governing. So to grow, enzyme juga, to protect, protein juga, to give structure and growth, diorang juga, defense pun diorang juga. So they are one of the key players, one of the important players to make living system works, right? So protein are polymers consisting of 20 kinds of amino acids, which all of these have different kind of their R groups. Okay, each protein falls into unique 3D structures defined by amino acid sequence. Okay, So how your protein fold depending on how amino acid is being arranged, okay, based on their interactions, whether it's attractions or repulsion. Protein structure is closely related to its function. Okay, so for example, the structures tu how it folded, concave shape ke, dia buat macam globular shape ke, dia buat macam spherical shape ke, all these are related to how it function in terms of structure ke, macam hydrophobic like cell wall. Okay, all this relates to its final function, to as a storage ke, as a transport ke, as a defense mechanism ke, as a enzymatic reactions ke, all this is related to the structures. Okay, habis dah. Done. All right. Okay, if organelles could talk, right? Really, I need a break. I need to feel quite fulfilled, whatever it is they're saying here. Okay, you can read on your own. Okay, you have any questions?
No.
Any questions? No questions? Really, no questions? My goodness. Senang ke lecture ni?
Ada kata tadi dah tanya banyak ah. So
Tanya banyak. Awak je tanya banyak. Semua orang faham Ilman, awak je tanya banyak. I'm just kidding with you. Kidding Ilman. Yes, Afia.
Just to confirm,
Yeah.
the whole idea of like the primary, secondary, tertiary, like primary is like the strand of DNA sequence,
Yep.
and secondary would be like the arrangement of them, they fold, they coil and everything. Like there are only two type of like secondary, one beta, one helix, one alpha
Yep.
helix. There's only two, like the interaction is just the folding itself. The interaction is just their own folding, like with the hydrogen bond and everything.
Yeah, only hydrogen bonds.
The tertiary would be like the matching, like they involve both helix and beta with the involvement of ionic, dipole, and
interactions when it comes easy for you to say, easy for you to remember. When it comes to tertiary, is the interaction of your R groups. Remember the R groups is different kan? Secondary hydrogen bond saja, which is between carboxyl and amino groups. Hydrogen bond, that's it. Either alpha or beta. Tertiary, now it comes forward another complex one, which is your R groups. Remember, your R groups had 20 different kinds of R groups, which is related to 20 different amino acids, right? So that is tertiary folding. Quaternary, finish it.
Quaternary will be the subunits, like they are bonded together with weaker bond, weaker interaction.
Yeah, weaker interaction, which most of that is hydrophobic bonds or hydrogen bonds. It's a combination of tertiary. Yes, betul. So 1, 2, and 3 is a primary, secondary, and tertiary. It's just one single polypeptide. Quaternary 2 is like different polypeptide comes together. Okay. Senang je. There you go. Afia dah sum it up dah. Habis. Senang je.
Just watch the video lah kalau tak faham.
Yes. I mean, that will be the basic understanding of how protein works. Okay. My next question will be, how this folding, all right, how this folding help you in terms of like, how do you relate all this with your food productions or anything with food? Can you relate this with food? Sama juga macam DNA. Protein is just DNA juga. I mean, protein comes from DNA. So how do you relate them? Try lah. Sama je, like I said, sama macam DNA je. Anyone?
Sir?
Yes, Rachel.
The misfolding of proteins, right? Like if we relate to food, like if there is a misfolding, like could there be possibility it changes like the size of that particular food or
Size.
something?
I'm not sure about the size. Possibly. In terms of a number of amino acids, it's more if you change if you in okay. Misfolding could be a lot of interpretations whereby instead you have instead you have this alanine amino acid, you change with phenylalanine. Okay, they just different amino acids. There's a possibility whereby instead you have 10 amino acid sequence, now you have 12. In terms of sizes, it depends, because if you have similar 10 to 10 but just different in terms of amino acid sequence, it won't change much in terms of sizes. Maybe it's bigger because maybe you have the interactions. Protein can be bigger, right? If you sort of like the interactions of all the proteins and interaction of all the amino acids were to be away from each other, so it become bigger group. Katakanlah like all of you guys, right? You guys, instead of you guys sitting close, you guys have how many? 40, like for example, 40. You guys tends to stay close to each other, rapat-rapat semua. Then you become smaller, right? What if one protein comes in, tukar dengan seorang, one person comes in, jadi mamat ni busuk ni, like really, he really smells like crazy bad. Okay, and he sits in the middle. So what happened to the rest of you guys?
Lari.
Dispersed, doesn't it? So now you become bigger group, and I say like bigger in terms of sizes. So correct me if I'm wrong, if the way you're asking, one misfolding could change its size. Yes, possibly, because the interactions. When it comes to interaction, it does not change the shape. It changes size. It changes the overall shape. It changes the function.
Or together with the functions also will change.
Of course, everything. It changes everything, every single thing. I can't stress you enough in terms of its functionality. Just remember what Pranazim has taught you guys: key and lock system. Okay, I punya kunci rumah will not fit your kunci rumah, right? Because it's just different. Although I have key, you have lock, I have lock, I still can enter your houses. Doesn't make sense. Everything is specific ways. One simple changes of the key, it just changes the whole thing. The whole changes the whole function, kacau or disturb your overall living system. Am I lagging?
A bit just now, sir.
A
A
bit
bit, okay. Am I lagging now?
No.
No.
Okay. So like I said, what I said just now, what did I say just now? Folding. Tak lupa dah.
Key house. Mostly. Everyone has lock and key.
Okay, sorry.
but-
Okay.
yeah.
Misfold. Yeah, misfold. For misfold tu, the list, if your misfold tu tak kacau, will not disturb your biochemical process, that's fine. Tak apa, dia tak kacau. Maybe it's be degraded, tak kacau orang. Tapi yang jenis yang kacau orang ni, protein yang kacau orang ni, kacau biochemical reaction. Instead of your biochemical reaction, from glucose supposed to make energy, and now your glucose tak make energy. Your glucose, I don't know, turn into something, I don't know, coagulate compound that can give you a certain bad disease or something. It turn your glucose into something bad. Instead of turn glucose to energy, it turns glucose into something very bad that relate your living system, makes you have certain diseases. That misfold is the one yang jahat. It gives a different reactions. Right? One example is your cancer lah. Easiest example. You know how cancer works, right? Mutation. We're not talking about X-Men, yeah. X-Men is mutation juga. But X-Men bagus lah. Maybe you can breathe fire, maybe you can produce ice, maybe you can go fly, or maybe you can, oh my god, if you can turn invisible, right? Or you read people's mind, then okay lah, best lah kan. But in reality, it doesn't work like that. In reality, mutation that we have is just mutation of cancer. That's it. About yang supernatural tu, I don't know, it's just all sci-fi. Okay, so how you know how cancer works, or how cancer give actually its bad effects? Tahu tak? I'm sure you know.
Isn't it like spreading it to the bloodstream and everything after like
That is
attacking
that
like
is the
one cell?
after effect of the cancer. You know what why cancer become cancer? What's the
Dia
main
jadi
features of cancer?
The cells don't stop growing. The cells continue to.
Yes. I mean, our cells always growing. For example, our skins. Our
But they
skin is
don't
just continuously growing every day. Cancer is the one that grow uncontrollably. There's no one guarding it. There's no one. I'm not saying when I say no one, just there's in your cell they have regulations, right? Control. Something or protein or some regulation that control its growth. It go when it's needed. Okay. If all this regulation does not exist within that particular cells, it start to grow uncontrollably, grow like crazy, grow makan, just keep on eating. If it grow, it's fine, grow lah banyak, and then become now it become benign. You know it's benign, cancer yang tak aktif. Macam ada you grow kat maybe your hidung or your telinga, something that's macam gumpal, just and then it won't affect any of your skin. That's called benign. It become dangerous when it become malignant, whereby the cancer grows so bad. The idea of cancer, it wants to grow, it has to consume a lot of nutrients. Where do you think it gets all the nutrients? The cancer cells,
Our body cells.
of course, your body cells. Okay, for example, in our class we have 40 people, right? Okay, and then in our class too, there are limited food. Every day you're gonna live for the rest of your life. Every day you're gonna you can makan three times a day, regularly. Okay, suddenly dalam 40 of you guys ni, 10 of you guys rebel. Tak kenyang makan 3 kali sehari, aku lapar lagi. Aku nak makan lebih. Katalah Ilman, siapa guys ni? Faris, Norman, kau ni makan kuat kan? Sally tak berhati. Aku makan lebih sikit, lapar, you nak supper, you nak dinner lebih. How's that gonna happen? You stole other people's food, right? Because you're just damn hungry. You wanna keep growing all the time. So when you keep stealing other people's food, so some of the girls, let's say Zakia, Balkis, Rachel, Afia, Sadi, instead of you makan 3 times a day, you makan 2 times a day. Some of you guys might makan 1 time a day. Some of you guys maybe tak dapat langsung. So what happened to you guys as you moving forward? Makan kena curi. So what happened?
Someone get food and die.
Becomes thinner.
Yeah, you get thinner, and along the way you what? Die loh. Because these guys keep stealing your food. Itu je korang ada. Basically itu je korang. These guys are growing. Bayangkan kalau diorang ni laki-laki ni, they are growing. Dia beranak lah tu. Ada dia punya anak-anak cucu cicit dia. Anak cucu cicit pun kau makan juga. Cancer, right? They are just continuously growing. And as time goes by, from these 10 people, they're outgrowing the 40 groups, and then the cucu cicit stole all your food. The rest of normal cells, you guys will die. So once once this guy killed the people, everyone in the class, what do you think happened next? They start to target other groups pula. Means they start to target other cells. So they keep stealing food. That's why cancer are dangerous and can lead to death, because they just grow uncontrollably, and then they just keep stealing food for other parts of the cells. So other normal cells cannot function. So still instead of the functioning, now the tak function, it just keep growing. Okay, when you have cancer liver ni, it won't function as liver. Now they just grow je. They won't serve function as liver. So that's when you have complications. Okay. Example, Ilman, for God's sakes. Clear? So in terms of your structures, like protein or DNAs, everything has to be specific. Okay. So that's similar juga when it comes to your foods. Okay. Any type of proteins that give certain structures or folding, that's the one that gives to a certain shape of your fruits. Like a mango is a mango, a banana is a banana. For chicken, that's how it's produce its own flesh. Okay, the meats, how it produces a certain aroma, how it produces certain shapes, the colors, everything is related to your DNA and protein. All this related to your functions. Okay. Sama juga nanti when you learn with Dr. Hafiz later on, you learn about how glucose is being digested, how protein is being digested, how fat is being digested, how your nucleic acid is being digested and processed within our body, so that we are growing at the same time. All other system, all other living systems is the same. Okay, so that's how you relate in terms of your proteins structures. All that is related to your the growth of your the how your food appears and looks like, and what it should be, and the characteristics. Okay, clear? Do you have any questions? So that's are sort of things that usually my favorite things to ask in the exams, I would ask what is structure of protein, or secondary, primary, tertiary, quaternary. That one I bagi my son jawab, dia boleh hafal. So I won't ask that kind of question. Maybe I ask some of them, but most of the questions would be critical thinking, your justification, and your understanding. Okay.
Sir, I have a
Hopefully it
question.
clears to you guys.
I have a question. Can you hear me?
All right. If no questions, okay. I did say that because now it's 4 o'clock. I did say that we should share the feedback inhibitions. Okay. Can anyone probably have another 10 minutes, that we can win our class. Can someone voluntarily share to us, to the rest of the class, about your feedback inhibition, feedback processes? Couple example of positive feedback and couple example of negative feedback, especially in food lah kalau boleh. Can someone share voluntarily, please? Anyone? Come on. Orang sama je. Mana yang tak on webcam ni? Yang mana tak on ni memang kena lah ni. Farhana. Baru on. Kena. Farhana, share please. Just tell me je. Just tell the whole class.
Wait, I actually have looked at the
You uploaded
examples.
Dah baca je yang upload tu. If you all submit, that's good. Submit je. You have ready until Friday. You can submit it.
I have looked for the examples. For positive feedback example is fruit ripening.
Okay.
So once ripe, it gives off a gas known as ethylene through its skin. When exposed to this gas, the apples near to it also ripen. So once ripened, they too produce ethylene, which continues to ripen the rest of the tree in an effect much like a wave.
Contoh buah apa?
Contoh buah apple.
Lagi?
Buah mangga.
Belasah je buah kan, buah mangga, buah apple, lagi buah yang ada. Yeah, more or less like that.
Apple is actually example kat sini.
Okay. Do you have any other examples?
Yang regarding to food, itu je yang that I could find. Like other is childbirth.
Childbirth. But childbirth is not polyethylene, right? That's okay. That is
no, I
positive.
mean like for positive feedback. Example.
Okay. All right, that's good. Yeah. What fruit? Banana. Apples and mangoes are the ones that have ethylene gases. For bananas, papayas, some of these guys, some of these fruit, the moment you cut them, then they start to ripen. That particular thing does not have ethylene gases, but instead they have a compound called polyethylene glycol, PG, if I'm not mistaken. Okay, those are the ones that ripen the fruits. Enzyme ni ada the ability of to macam you punya colors, right? From bananas, the colors, it change the colors from green colors, change the pigmentation from green to yellow colors. At the same time, you punya bananas punya flesh tu, if you makan banana yang tak masak, kelat and pahit, right? So your ethylene glycol has the ability to break down the saccharides of your bananas, make them loose, at the same time release sugars. So that's why bila makan banana masak, banana tak masak, you have sweet and nice and juicy for your ripened banana, and unripened banana is kelat because the punya sugars has not been released. Faham ke? Yep.
Faham.
Okay, good. Thanks. Yeah, Iman. Thanks, Iman. Alright, someone else, please. Iman, nak ke? Do you have something?
Nak kata.
Cepat. So who is it? Iman or anyone else? Cepat. Come on. I don't have all day. Mana yang tutup video cam lagi? Memang nak kena ni. Mari saya cari siapa yang tutup video cam lagi. Mariam. Where are you, Mariam?
Hi, doctor.
Hi, what happened to you? Tak mandi lagi kah?
No, the room is dark.
The room is dark. What are you doing in a dark
No,
room? My goodness. We are in the 21st century, Mariam. I think all houses have lights. I think your parents will think what sort of dodgy stuff you do in the room, in the dark room. I have online class, Mom. Thank you so much. Okay, cepat, Mariam. Example list.
Does it have to be related to food?
Possibly, yes. If you can't, you have to justify if you can't.
I was
Yesterday,
looking...
I got 2 people telling me childbirth and menstrual cycle. My goodness. Okay lah, I can accept that.
I got positive feedback, but it's for blood clotting.
Blood clotting, all right, why not? Go ahead.
Okay, so basically, like when the tissue is like torn or injured, a chemical is released. The chemical like causes the platelets of the blood to like the platelets in the blood to activate. So once this platelets has like been activated, they release another chemical signal to like more until the wounds like clotted up, so like no more blood gets released.
Right. So more. So is it feedback or positive? Sorry, is it positive or negative feedback?
It's a positive feedback.
Is it positive? The more
in context where when you injure yourself, so you don't lose blood.
All right. Positive means the more blood come, more. Okay. I thought it's supposed to stop. Is it negative? No, yeah.
The internet.
So the injury is the platelet release, right?
Yeah.
So more platelet is being released. All right, yeah, makes sense. Okay. Okay, thank you. Lagi? Siapa yang lagi senyap dalam kelas ni? I got probably like 2 more. Now we have 2 positive, probably negative perhaps. So who wants to share negative? Tak maulah macam high school again. Volunteer lah sikit. Come on lah, be more berani sikit. Takut dengan malu dengan whole class. Apa? Ada crush korang ke? Come on guys. Dia tengah Google ni. Obvious sangat kot mata tu baca. Tengah Google kan, mana ni buat negative feedback. Tak buat lagi ni, tak submit lagi ni. Cepat. Fitri. Suddenly I caught my eye on you. Fitri. Yes, Fitri. Using your phone ke, Fitri?
Yeah.
Alright, come. What is it?
Tengah bunyi kipas tak? Sekejap eh. Sorry.
Huh? Tak dengar lah.
Okay. Saya dapat yang feedbacks yang tak berkenaan dengan foods.
Why not? Boleh lah. Tak ada berkenaan dengan food ke korang ni? Banyak je food kan?
Kalau yang ATP productions ni boleh.
Yeah, why not? ATP production, yeah, can.
Sekejap. Okay. ATP Alamak. Saya baru dapat ni kejap eh. ATP is from glucose. There are a series of enzymatic reaction in our cells.
Okay.
Glucose is a stable form of energy currency which can be absorbed from food or transported around the body as needed. ATP, on the other hand, is unstable and will spontaneously lose its energy if it sits around unused. So, after the reason, it's important to regulate the breakdown of glucose and the production of ATP. Producing too much ATP results in energy loss, and glucose depletion could mean big trouble in consumptions where food is scarce. To control the amount of glucose that is broken down to produce ATP, the first enzyme in this breakdown chain is regulated by ATP. If ATP bind to the enzyme, it will not break down further glucose.
Dah.
Dah.
You faham tak apa yang baca tu? Exactly. Alright, Fitri, so be careful. Yeah, I know you're not ready. Okay, so be careful. I don't think you understand what you're reading, right? Okay, so be careful. Alright, thank you for the sharing.
Sorry.
Can I get two more negative? Afia, do you have anything?
Actually, I do, but I haven't exactly arranged the word. I'll try to.
It's fine. Yeah. All I need from you guys is just layman's term. I don't need.
Okay.
I don't need. Is that copy paste from the internet? As long as we understand it, it's fine.
Okay, it's actually from the process of digestion.
Okay.
It's the regulation of gastric juice. The negative feedback is the inhibition of gastrin-induced acid secretion by the intestine-derived hormone secretin. It's like the enzyme from intestine gives the inhibition of secretion of the gastric juice. It's like the hormone from intestine prevents the secretion of gastric juice.
Oh, okay. Why?
It's a deregulation in the gastronomic, like the process. It's like to maintain the acidity and this thing.
Yep.
Yeah.
To maintain the acidity within one stomach, because once intestine enzyme is being produced, meaning that your food is moving from your tummy towards your intestines.
Yeah.
So you cannot afford to produce more acids, because if acid is not allowed, it's not allowed to get into the intestine. That's why you have bad digestion. Okay, there are people when you have bad digestion is when your acids go through your intestines. Then when you're gonna have different kind of problems lah. Okay, all right, that's called negative or positive?
Negative, because it's the inhibition of the other one.
Okay, good. Okay, last one. Okay, why don't you, Rachel? Do you have anything?
Doctor, can mine's not like, it's not related to food lah somehow.
Can lah, if you digest about food ke, makan food or anything lah, it's fine.
Mine's about, was blood glucose level told just now? No, right?
What?
For negative feedback, right, sir?
Yeah. Could be positive.
Okay.
Whatever you have.
I got regulation of blood glucose levels.
Okay, yeah, right. Go
Okay,
ahead.
so basically, just to summarize, insulin, which is produced, it will deliver a signal to the liver, muscles, and then other cells to store those excess glucose when our blood sugar level increases. So, however, some of that glucose will be stored as glycogen in the liver and muscle, while the rest will actually be stored as body fats. So that's negative feedback inhibition.
All right. Yep.
Where
Good. Thank you. Yeah, positive. Dah, tu je.
I got positive for digestion, sir, ripening in fruits, and blood clotting also.
All right, same. Okay, never mind. So I expect you, the rest of you guys, to submit your feedback inhibition in class, in submissions. I already create the submission link in your Putra Blast, right? So submit in there. Like I said, please do not copy and paste or plagiarize. Yeah. So I can really tell if you take it from internet, it's just kalau cantik sangat dari internet, memanglah itu. Memang you are doing from internet. So please do not do that. Yeah. Explain in your form in the most simplest way that you can understand. I can definitely understand. Your grandmother can understand. Simple je. I don't need that mechanisms. This react with that will turn into that. Oh, I don't need that. Just simple term. Okay. Same goes for your GMO or CRISPR genetic modification this Friday. I don't need the mechanisms. All I need is just simple definitions. If you can provide the figures, I would appreciate if you can have your PowerPoint. Right, I've called randomly. I think about 1 hour. There's a lot. There's plenty people can present. Again, I will choose randomly. Put in PowerPoint slides. You can have your figures and everything. You can talk about the figures. You present it as well. And then as well, explanation too, you can explain on your own. Present ya, not read. Jangan baca. Baca sikit-sikit whatever you tulis tu boleh lah, tapi taklah baca macam what exactly in the internet. Jangan, okay, please jangan, I warning you from the get-go, jangan on that particular day, if I will randomly pick you guys, baru tu hege-hege nak Google at that time. You'll be in trouble. Okay, so please, I don't expect that to happen from you guys. And you all are good students, please, so prepare. Tak susah pun, I think within less than 1 hour, just can finish it. Right, any questions before we can end our class? No? All right, if not, thank you very much. That will be our last lecture. I'll see you on Friday then. Okay, Mariam, you can turn off your lights now, Mariam. Kesian, Mariam. Sorry,
Doctor.
Yeah.
Yang explanation tu, does it have to be an essay or something? Kena panjang ke?
It doesn't have to be essay. It doesn't have to be a point form. It's just kalau you nak buat essay boleh lah kalau you buat in your own words, tapi janganlah copy and paste. I don't expect like one whole page of essay. Simple je. Macam some of you guys punya classmate ni cakap tadi. Dah, polyethylglycol ethylene is just gases spread around that net, and then this is what sort of feedback, positive and negative. Simple je. Okay. This assignment, yeah. So make sure this all have marks, yeah. So if you can't do it and then if you don't present well, that just affect your marks. Okay. So if you have good marks, just do it well. Any question, guys? If no, thank you very much. All right. So have a good day. I'll see you on Friday. Bye-bye.
Thank you,
Thanks.
Doctor.
Bye-bye.
Thank you, Doctor.
Thank you,
Assalamualaikum, Doctor.
Thank you, Doctor.
Thank you, Doctor.